Your latest Murgitroyd webinar will start in a few moments. For more information on today's panel and our company, visit murgitroyd.com/webinars.
Hello and welcome to the 8th series of Murgitroyd online seminars. We are delighted to reconnect with our regular listeners and a warm welcome to those of you tuning in for the first time. Our webinars are here to keep us all up to date on the latest developments in the world of IP. Please don't forget you can submit questions via email at webinar@murgitroyd.com. You can also contact us by Twitter using hashtag #MurgitroydWebinars. And remember to tag us at Murgitroyd. So, let's go straight into today's webinar.
Hi, I'm Rae. I'm a patent scientist here at Murgitroyd, and I'm delighted to be joined by Cath Coombes. Hi, Cath.
Hi, Rae.
Cath is a director in our patents group based in York and specialises in CRISPR technology. I mean, you have at least a decade's worth of experience in working with this technology from patent prosecution through to being involved in patent disputes. And I understand as well that you have provided comments and peer-reviewed journals such as Nature, The Scientist, and more recently in IM. Is that right?
Yeah, absolutely. Yeah, I mean, I find it a very interesting space, particularly since I've followed this field from the use of CRISPR technology as it is in nature, which is part of an adaptive immune response of a bacterium, to see it evolve through into gene editing. And it's been quite fascinating to see the development in this field.
Yes, it is certainly an exciting and promising technology, and it holds so much potential in medicine. It would be wonderful to learn more about what CRISPR is and how it developed, because as you say, it has been known in bacteria who've used this means to fight against viruses.
That's where the term CRISPR actually originally came from. So back in the 80s, they were looking at a bacterial genome, and there's with these repeated sequences, and they were always set about the same distance apart, and they were clustered together in the genome. And whilst it's a misnomer for the most well-known type of CRISPR system, the P part stands for a palindrome, because it was a palindromic repeat. So they're clustered regularly interspaced palindromic repeats. That's what CRISPR stands for. And it piqued the interest of scientists, because why do we have these repeated sequences clustered together in a bacterial genome? And I got into the field because it was discovered that this was part of a bacterium's innate adaptive immune system. And one of the key findings that led to that is the realization that the bits of DNA in the genome between these repeats were actually corresponded to little sections of phage DNA. And so the sections between CRISPR repeats in a bacterial array are called spacers. And they were the complement to a protospacer in a bacterium, which is where we get one of the other key terms in the field, the PAM motif, a protospacer adjacent motif, because it was really important if you're utilising the recognition of that section of phage DNA to understand that a bacterium's being invaded and it needs to be cleaved. But the bacterium itself can distinguish between the phage and itself in its bacterial genome. And that's the role of the PAM motif in bacterial immunity, because that PAM motif is important for binding and for the Cas enzyme to undergo the kind of conformational changes that's required to access the phage DNA to its endonuclease domains to cleave the phage DNA and hence provide immunity for a bacterium. So I started in the field, and it was all to do with utilising these aspects to improve food production. Because obviously, if you're producing certain strains of yogurt, for example, and you've got key bacterial strains you want to use, you don't want them compromised by phage attack. So if you can modify that bacterium to have increased resistance to phage, that was very beneficial. And they were some of the early patents that I prosecuted and then went into opposition for, starting maybe 2007. So yes, it's been a fascinating journey seeing food technology aspects, and out of nowhere, for me personally, seeing this field explode into a fantastic mechanism to be able to access DNA within plants or animal cells in a very easy way compared to previous technologies.
It is really wonderful because it has such precision in being able to locate a specific sequence of the DNA in order to edit or change that part. It would be lovely to learn how that is achieved.
So this is what is such a huge leap with this technology. I mean, if we look at the existing gene editing technology we had prior to this, each time you wanted to access a certain location within our DNA, you'd have to decide where you wanted to bind a protein to, but then you'd have to synthesize the entire protein for it to be able to bind to that particular region. And to obviously construct and develop a protein takes a long while. So whilst CRISPR technology is great with the nuclease aspects, which people tend to focus on when we talk about cleaving DNA and editing, it's really its ability to get to specific areas of DNA that we're interested in quickly, which is, for me, the mind-blowing aspect of this technology. And the difference here is the protein stays the same. So the Cas enzyme stays the same, but it interacts with a relatively small piece of RNA. And it's an even smaller portion of that RNA, about 20 nucleotides, that needs to be changed to relocate it to a different destination, shall we say, within our genome. So it's a readily reprogrammable DNA binding protein, because to synthesize a short section of RNA can be done very, very quickly. That's not to, of course, suggest that it's necessarily straightforward working out exactly where you want to go and how to optimize that to reduce off-target effects, by which I mean accidental binding somewhere else in the genome where we don't want it to be. But the synthesis itself of that piece of RNA is very quick compared to having to completely design and synthesize an entire protein.
Certainly interesting to learn. What is special about Cas9? We have heard so much about Cas9 recently, but why specifically Cas9?
So first of all, CRISPR systems can be put into two classes, class 1 and class 2. Now, what's interesting about class 2 CRISPR systems, of which Cas9 is a class 2 type 2 CRISPR system, is that you only need one Cas enzyme involved in this, what we call, from the bacterial terminology, interference aspects. But that basically means in order to carry out the binding and the cleavage, you only need one Cas enzyme, whereas the class 1 systems actually are multiple proteins involved in that. And of course, if you have multiple proteins involved in that, you have more difficulties getting them to the correct location, interrelation with one another. Whereas if you've got one small Cas protein, it's easier from delivery aspects, and it's much more straightforward in order to be able to utilize it from gene editing. And Cas9 is a type 2, class 2 CRISPR system. And when this technology moved into gene editing, it was the only known class 2 type CRISPR system. But we've seen massive developments in the field since then. So we now have type 5 and type 6 systems, for example.
And have these proteins been modified, which is why we have the different classes now? Or are they simply looking at different types of protein now?
Yeah, so the classification, as I said, the class 1 to class 2 is based on whether you need a number of proteins involved in a combination of binding and cleavage, or as opposed to a single. And then there are differences depending on, for example, can it act on RNA rather than double-stranded DNA? It is one aspect that differs. So Cas13 acts on RNA. Cas12a acts on DNA, but instead of, as with Cas9, creating a double-stranded break, if you don't deactivate the endonuclease, it will create a staggered break in the DNA.
And moving forward, where do you see this technology going? For instance, which types of diseases is it specifically used to treat or prevent? Or does it have a much broader application?
It has applications everywhere, frankly. Oh, wow. So in industrial biotechnology, imagine you can modify a bacterium and its DNA. Well, that's fantastically beneficial if you want to use that bacterium as a factory to produce a chemical that's desired, because you can modulate its pathways to synthesize a particular chemical. So it's great uses in industrial biotechnology. And then when we think about agricultural applications, for example, we know we want to increase drought tolerance, we want to increase yield. And by having readily access to the genome of a plant cell, it's fantastic for basic investigation as well. So not only do we, there's various modifications that we can make, which we know are going to be beneficial, but there are so many genes that we don't actually know what the function are at the moment or how they interplay with other genes. So what happens if we interfere with that gene expression or we activate particular genes and what the consequences are. So very basic science in kind of understanding more about the mechanisms of action and interplay of genes. CRISPR technology is fascinating for that. And then we can go on to human diseases. And, you kind of think of so many diseases are caused by single nucleotide polymorphisms. So that's where we compare a healthy individual with one exhibiting a particular disease, and it's resultant to one base change in their genome. And when we know what the error is and we know what the correction should be, then theoretically we can make that correction. And the field has, again, moved on a lot since its beginning. And there are various base editors now. So you can attach to Cas9, for example, another protein called a base editor. So that tows the base editor to the correct location. And instead of using Cas9 to cleave, you use the base editor, which will substitute out a specific base and replace with another specific base. So really fantastic technology. And given we're in a pandemic, unfortunately, which is where we're recording from home, another amazing use of CRISPR technology is to do with detection. So Cas12a, for example, when that was first developed, it was also called CPF1. I discovered that in the presence of a particular target sequence, as designed, it would cleave. But then it would go on and have this other activity where it would go around and cleave other bits of single-stranded DNA. So that's fantastic for diagnostics, because if you imagine you design the CRISPR RNA so that it will bind to COVID-19, then that enzyme will go off and cleave other single-stranded DNA, which you could put in with some fluorescent marker, for example. And therefore you can develop a way of detecting COVID-19. And in fact, Sherlock Biosciences has got emergency use authorization from the FDA for a COVID test using CRISPR technology, which, given that, Cas12a was only discovered less than 5 years ago now, 6 years ago, it's amazing. It's absolutely phenomenal progress. For me, it's the ability for it to be reprogrammed and to be directed to bind, which is really, in essence, the amazing thing about CRISPR technology. Inherently, Cas9, Cas13, Cas12a, various Cas enzymes have the ability to cleave, but we can easily turn this cleavage ability off. So, for example, we might want to just prevent Cas9 from cleaving both strands, but to just nick strands, which will be useful in generating repair mechanisms within a cell. Or you can completely prevent the endonuclease systems from working, so it just binds. Now that can cause interference, CRISPR interference, but you can also use the Cas enzyme a bit like a tow truck to tow other tools there. So earlier I talked about a base editor, to tow a base editor to the right place so you can switch out specifically. But there are various different tools that you can do depending on what you're looking to achieve. And so you can think of it as a bit of a tow truck being towed to the right location with various tools loaded on.
Yeah, that's a very good analogy, actually. It's easy to understand. So from a patenting point of view, how easy is it to patent in this area?
I think you need to kind of consider what you are patenting. So as I said, this is such a basic research tool now for an understanding of various abilities that there'll be lots of information disclosures that you get from universities or from clients which will mention the word CRISPR and use CRISPR. But a lot of those inventions won't be for the CRISPR technology. It's just a technique, a bit like PCR, that was used in the process, and the invention didn't lie in CRISPR technology. And so for those, standard patentability applies. where we're looking and our invention lies in the use of the CRISPR technology itself. Obviously, this field has grown massively. I mean, the last I heard, there was over 6,500 patent families for CRISPR technology. So we're talking huge growth since 2012, when the earliest patents for gene editing were filed. So we have various considerations that we need to think about. And I think it really helps if you have somebody who has a good understanding, not just of CRISPR-Cas9, but of the other CRISPR systems, how they work, and really ensuring that you're claiming correctly depending on your invention. Because, for example, if you're just looking to have a particular enzyme to tow to the correct place, and then your invention's on what it is towing, so the toolkit, as I said earlier, then it doesn't have to be Cas9, because Cas9 is one example of a class 2 system. So you wouldn't want to be drafting to a CRISPR-Cas9 because that would be too narrowing. A deactivated Cas13 or Cas12a or Cas12b, up to I or further by now, would work, as long as it's deactivated, so it's not doing that cleavage. So it's important that there is a good understanding about how the different CRISPR systems work, and you're not either accidentally narrowly claiming or accidentally overclaiming and encompassing things that wouldn't actually work.
It would be very interesting to get a better understanding about the decision by the European Board of Appeal with regard to Broad. Are you able to share more?
Yeah, absolutely. So in 2011, it was first discovered that type 2 CRISPR systems worked a bit differently. So at the beginning of our conversation, I said there's a bit of a misnomer about having it being palindromic. And that's because for a type 2 CRISPR system, it doesn't hybridize to itself to form a secondary structure. Instead, CRISPR RNA will hybridize to a tracer RNA, and that tracer RNA element was discovered in 2011. And then in 2012, this was when it was first being considered in the journals that potentially type 2 CRISPR systems could be used for gene editing aspects, whether that be in bacteria or in higher organisms such as plants or animal cells. And very surprisingly, given such a leap for this technology, there were actually 6 different players who kind of got there about the same time. And Broad, their first initial filing was on the 12th of December 2012. Sigma Aldridge filed 6 days earlier, on the 6th of December 2012. A South Korean company called Torgen filed in October, so a couple of months earlier than that. And UC Berkeley filed as early as the 25th of May 2012. Broad's strategy was actually really good in a number of aspects. So they discovered earlier on that, they could see the fundamental impact of this technology, and they decided to expedite prosecution and have a thicketing approach. So from their earliest priority filing, they filed no less than 10 PCTs on the same day, claiming priority. And these were expedited, so they entered the US very early, they entered Europe really early. In fact, so early that a couple of their patents were granted before UC Berkeley's, which was filed months earlier, even entered Europe and was citable against them. So they ended up in a really strong position. Sounds great so far, doesn't it? But unfortunately, the Board of Appeal revoked their patent and several patents since of their key group of patents in Europe on what some people would call a technicality, and that's that the formal requirement for priority wasn't met. Now, the earliest priority filing for Broad had 6 inventors, and under European patent law, they're considered a joint applicant, and for you to file a later application claiming priority, it has to be by those joint applicants together, or they have to be a successor in title. So by that, it's meant that they have to have had permission in order to claim the right of priority. In this case, the earliest filing was made, and after that, as is quite usual actually in the US, the inventorship determination occurred after that initial filing. And it was decided, as I said, that there was various different inventions in this earliest filing, which led to numerous PCTs being filed. But for eukaryotic aspects, it was decided in this inventorship determination that Luciano Marafini, who was one of these initial inventors, and his institution, Rockefeller University, didn't materially contribute to CRISPR technology in eukaryotes. So he was no longer named as an inventor in a number of the PCTs, in fact, all but one. Yeah. And his institution, Rockefeller University, wasn't named as a joint applicant. In the US, Rockefeller University filed a continuation on a granted patent by Broad and MIT, adding themselves as a co-applicant, and Luciano Marafini as a co-inventor. And it was laid bare on the record in declarations that following this inventorship dispute, there was silence with Rockefeller University, and these PCTs went on to be filed. So it was clear from the declarations that were put on file that there was no permission from Rockefeller University or Luciano Marafini to claim that right of priority. And it's because of this that the smaller group of co-applicants were considered not to be a successor in title, so they're not entitled to the priority date. What does that matter? I can hear you'd ask. And in a lot of fields, it wouldn't matter that much. But when you've got a load of players in a field at the same time, and you've got lots and lots of publications coming out in a very short space of time, it matters an awful lot. And it certainly matters when their own publication came out prior to the filing date. So that was Kong 2013. So their own data, so it's identical, is now prior art against them. And so that's led to some revocations in Europe, and it was upheld at the Board of Appeal. But some other cases, for some other of their technology, actually there were additional inventions within that which didn't need that right of priority and were okay. So unsurprisingly, these days, there's lots of collaborations between institutions. And one of the things with collaborations is, a boundary is put in place beforehand as to who should own the technology, who should have a right to file on that technology. In hindsight, if agreements had been put in place prior to that initial filing, the decision wouldn't have happened for Broad, because were it not for that, then it's highly likely that their patents wouldn't have been completely revoked in Europe, and they would have had a lot stronger rights in Europe.
Yes, it's certainly not a situation you want to be in, given how lucrative this area is, isn't it?
Yeah, absolutely. And it's one of those things that I spent quite a while scratching my head over, because I kind of put myself into the shoes of the US attorney who did that inventorship analysis. And it's a really difficult place to be, because what do you do when there is a dispute, even if you wanted to solve the issue? And with hindsight and a lot of thought, the only thing I could think of would have been to have filed one giant PCT with all of the inventions in the claims, and then to have divided that out. And as you divide it out, to drop inventors and to drop applicants, because then your PCT would have had that succession in title.
Yes, I see. So it is very important that in any collaborative work that you establish that chain of entitlement early, and you also ensure all your agreements are in place, right?
In an absolute ideal world, I would completely agree with you. But I'm sure if you speak to anybody in university tech transfer, they would be smiling at that comment and saying, yes, wouldn't that be lovely? I guess, one of the things that we could learn from this is that it is useful to, keep records of your collaborations, your contributions, because, as I said, in this case, the dispute came about because Luciano Marafini and his institution felt that they contributed more to the eukaryotic aspects as well, but they didn't have the proof to put before the US attorney to be convinced that they should be named as an inventor, etc. So perhaps if they had that paper trail, then it would have been easier to determine.
Yes, that is certainly very informative of what can go wrong and the steps we should take to avoid such pitfalls, as you've just discussed, such as the claims to priority and entitlement, and what they can put into place if possible. And that can sometimes be quite difficult. But alongside this, as you've already mentioned, there are over 6,000 patents in the area So it's quite heavily loaded. How can we help the applicant navigate themselves such that they get the protection that they need and they're able to market their product and build their business?
So I guess your question's covering two aspects there from what I understand. So first, the patent side, but also how to navigate this somewhat complex landscape. And I would describe it definitely as very complex when we're talking about the type 2 CRISPR systems, because there's big disparities between jurisdictions as well with regard to this technology, which adds to the complexities. So I guess from the patenting side first, as I mentioned earlier, I think, the heart of any initial assessment is, what is the key invention here? Is it the CRISPR technology itself? Is it the use that the CRISPR technology has been put to? And once we've made that kind of determination, then I think, it's a case of getting some very good searches. In terms of navigation, this can go hand in hand. So, for example, if you want to go into a particular area and do research in a particular area, getting engagement with a patent attorney in the field early on can be beneficial, because as well as considering the patentability aspects, we can marry that with potential freedom to operate aspects and consider actually, whether developmentally you'd like to go down a slightly different avenue, which isn't so patent heavy and complex, should you get through to the stage where you're obviously looking for freedom to operate. I think a great assessment is to look at these hand in hand. So from a patentability perspective, yes, we want broad technology, but from a way to navigate the landscape, is the focus of research, should we be looking at some of the newer CRISPR systems, or, for example, the Cas12a landscape, if the Cas12a is applicable, is less complex compared to Cas9. If we're looking into stuff which has to do with delivery, then actually should we be narrowing down to those CRISPR enzymes that are particularly smaller and navigate ourselves away from some of the earlier patent issues? But it really depends on field as well. So there's some good collaborations in place with regard to agriculture right now. And we're seeing quite a bit of an attempt to have more of a clearing the way when it comes to reagents, for example, and co-licensing between parties. But at the moment, when it comes to human therapeutics, we don't really have that in place. There has been calls for a patent pool, and MPEG-LA have been involved in that for a good few years now. But whether that actually comes to fruition for human therapeutics, I'm always a little bit reticent about, because obviously of the exclusive nature that's required in order to get the investment for clinical trials. I'm not sure that necessarily a patent pool would be the most appropriate way to navigate the landscape for human therapeutics, but certainly for other areas, we're seeing more collaboration, and I hope that would continue, because otherwise it is quite a tricky landscape to navigate.
Thank you very much, Cath, for such an informative discussion. I certainly learned a lot, and I look forward to speaking to you again on this topic. I also understand that you have recently written an article for Murgitroyd, which is on the Murgitroyd website, and you've posted on LinkedIn regarding the patent development in this area in the US. Could you tell us a little more?
Yeah, absolutely. There's been a recent decision on the second patent interference over who's first to invent between two of the six parties, so between UC Berkeley and Broad, for CRISPR-Cas9 technology using single guide RNA in a eukaryotic environment. And if anybody's interested in that, then please get in touch and I'll happily send you a copy.
Oh, thank you so much for your time again. I look forward to our next talk.
Oh, thanks, Ray. All right, take care.
Bye.
We've reached the end of today's presentation. To find out the topic of our next webinar and to register, visit murgitroyd.com/webinars. And whilst you're there, why not keep up to date with our latest content by subscribing to our mailing list? Thank you for your valuable time today and for all your questions. We hope you found the subject matter relevant and well explained. Should you have any other questions or wish to discuss the topic further, please contact us at webinar@murgitroyd.com or via Twitter @Murgitroyd. Until the next time, goodbye.
Our business is founded on trust and collaboration. The subjects we choose for our webinars are topical and informative. We are always looking for suggestions for new topics for discussion. So if there's an area of IP that you are keen to understand better, please drop us a line at webinar@murgitroyd.com.